Assalamualaikum and salam sejahtera. So my name is Alia Nabila binti Sarimin and my matrix number is S54890. So in this video, I will discuss the flow from traditional to modern understanding of diversity marine phototroph. What is a phototroph? A phototroph is the organism that can use visible light as a primary energy source for their metabolism, which is a process known as photosynthesis. What is a photosynthesis? Photosynthesis is a process where a green plant and some bacteria will synthesize their nutrients from carbon dioxide and water by harnessing the light energy. So there is two types of photosynthesis. One is oxygenic photosynthesis and the other one is anoxygenic photosynthesis. So in oxygenic photosynthesis, the light energy will trans will transfer the proton from water to carbon dioxide to produce carbohydrate and oxygen as a byproduct, while in anoxygenic photosynthesis, it will use a proton donor except from water, and it also does not produce any oxygen as a byproduct. So there is three types of phototroph. The first one is photoautotroph, the second one is photoheterotroph, and the third one is chemoheterotroph. So photoautotroph and photoheterotroph both are still using light as their source of energy. But then photoautotroph are able to synthesize their own food, while the photoheterotroph depends on organic matter that are already produced by other organisms. The third one is chemoheterotroph. This chemoheterotroph is commonly found in the deep water area where there is a hydrothermal vent. Chemoheterotroph organisms obtain energy by the oxidation of electron donors in their environment. It uses inorganic energy sources to synthesize organic compounds from carbon dioxide. So the next one is, this is the timeline in the ascending of marine phototroph. So this one started from mid 1970s until 2011. So in mid 1970s, they only thought that diatom and flagellate is the major primary producer in marine environment. In late 1970s, the unicellular cyanobacteria Cyanococcal species was revealed. And in late 1980, a cyanobacterium named as the genus Prochlorococcus also revealed by using high-performance liquid chromatography (HPLC) and cell detection technique using flow cytometry. In 1996, a Cyanobacterium acriolaris marina was reported to use CH chlorophyll D instead of chlorophyll A. In 1998, a Cyanobacterium-like particle CuCyNA was detected. In early 2000, two significant findings were reported, which are aerobic and oxygenic photosynthetic bacteria (AAPB) and proteorhodopsin-containing bacteria. In 2007, a phytoplankton that does not belong to any known phyla named Picobellephyte were discovered by metagenomic analysis of plankton fraction less than 3 micrometer in size. In 2010, another redshifted chlorophyll F was reported, and a new eukaryotic organisms that belong to Haptophyta were discovered by pigment composition analysis. And in 2011, the new candidate phylum named Repermonads was detected by metagenomic analysis targeting plastic DNA. So the first one is the traditional understanding of primary producer. In mid-1970s, only the atom and flagellates were known as the primary producer in the marine environment. This is because the phytoplankton is widely distributed in the mid-range water quality, in the wide-range water quality, including eutrophic and oligotrophic environment. Eutrophic environment is the environment that are rich in nutrient, while the oligotrophic environment is environment that less nutrient. So these diatom and flagellates are the major organisms that can be collected by using the plankton net at that time. So the diatom and dinoflagellate is algae. Diatom is a single algae, while dinoflagellate is a unicellular flagellated algae. So diatom is the algae that live in houses made of glass like this. So it is the only organism on the planet with cell composed with transparent opaline silica, while the dinoflagellate possesses one or more flagella, which are long tapering hair-like appendages that act as an organelle of locomotion and feeding. Why did only these two is recognized as the primary producer at that time? It is due to the approach of phytoplankton sampling and the methods of phytoplankton detection at that time. At that time, they only used plankton net with mesh size 100 micrometer, 70 micrometer, and 60 micrometer. That is why only diatoms and flagellates were trapped inside the plankton net. But then, with the development of technology in study of phytoplankton, it shows that there is a lot of predominant phototroph in our marine environment, but in smaller size. In late 1970s, unicellular cyanobacteria Cyanococcus species were revealed to be predominantly distributed in the marine environment, especially in tropical and subtropical regions. It is a very tiny cell, so it is called as a picocyanobacteria. It plays a significant role in the marine environment as a primary producer. The next one is in late of 1980s, a new picocyanobacterial was recognized and it is named as the genus Prochlorococcus. A Prochlorococcus contains divinyl chlorophyll A and it does not contain phycobilic pigments which are common in Cyanococcus species. So this Prochlorococcus perform oxygenic photosynthesis using DVCHL A/B, which can efficiently absorb the blue light dominating in the ocean. It is dominant phytoplankton in tropical and subtropical regions between the latitudes of 30 degrees north and 30 degrees south. At several regions, the amount of DVCHLA is 3 times higher than chlorophyll A. That is why Chlorococcus play a significant role as a primary producer in the marine environment. So the discovery of ubiquitous photoheterotroph. Ubiquitous means found everywhere, where heterotroph is an organism that depends on organic matter that are already produced by other organisms for its nourishment but still obtain energy from the sunlight. In early 2000s, there are two significant findings were reported, which are aerobic and oxygenic photosynthetic bacteria and a proteorhodopsin-containing bacteria. The aerobic and oxygenic photosynthetic bacteria is distributed widely in euphotic environment and grow heterotrophically. It is derived from anaerobic and oxygenic photosynthesis bacteria ANAPB. This AAPB contains bacterial chlorophyll BCHLA, a photosynthetic pigment in aerobic and oxygenic bacteria ANAPB. However, AAPB is an aerobic organism. It requires molecular oxygen for their growth and BCHL and bacterial chlorophyll A synthesis. The growth is faster than other heterotrophic bacteria in the seawater. So it acts as a primary producer in the marine environment. The second one is proteorhodopsin-containing bacteria. These bacteria contain proteorhodopsin, which is the rhodopsin-like protein. One rhodopsin-like protein was known to contribute to adenosine triphosphate ATP synthesis. Rhodopsin is very sensitive to light. It still unable to obtain light at low light intensity. So rhodopsin-like gene were detected in seawater in the form of E. coli, and it acts to form transmembrane proton electrochemical gradients. This proteorhodopsin-containing bacteria plays a significant role in light energy reaction in marine environment. On the other hand, proteorhodopsin-containing bacteria also plays another role, which is it can be an advantage when we are in starvation. The next one is oxygenic photosynthesis using far red light. The photosynthetic pigment chlorophyll A will absorb light from visible light spectrum. Oxygenic photosynthesis using far red light. In 1996, a cyanobacteria Chloris marina was reported to perform oxygenic photosynthesis using chlorophyll D. Chlorophyll D is a shifted chlorophyll. It absorbs far red light almost 30 nanometer longer wavelength than chlorophyll A. They use chlorophyll D for light harvesting antenna and for chlorophyll in the reaction center. These cyanobacteria perform oxygenic photosynthesis and widely distributed as epiphytes on seaweed. So the second one is in 2010, there is another red-shifted chlorophyll F were reported. It is from a cyanobacterium isolated on microbial mat on stromatolite. It has absorption maximum around 720 nanometer. But then these two cyanobacteria was not counted as a marine as a primary. But then these two cyanobacteria was not counted as a primary producer because there still need a lot of research to determine how their works and how their activities. So the next one is the discovery of eukaryotic phytoplankton in 1990s to 2011. Until early 2000s, only dozens of species in Chlorophyta, Heterocontophite, and Haptophyte were known as eukaryotic phytoplankton. But then until 2011, there are three non-prokaryotic phytoplankton were reported. The discovery of pico-eukaryotic phytoplankton around 2010, several reports on significant amount of picohaptophytes were published. A carotenoid 19-hexalloxyfucoxanthin 19HF was predominantly detected. 19HF is a specific pigment to the algae in haptophytes. Picohaptophytes contribute significantly to primary production in the marine environment. This is because haptophytes contribute 50% of the total number of phytoplankton, and then the picohaptophytes contribute 5% of the total number of picophytoplankton. So the next one is the discovery of pico-eukaryotic phytoplankton. In 2007 and 2011, two algae with unknown phyla were discovered. Both were discovered by metagenomic analysis. In 2007, the phytoplankton was named Picobeliphytes. It does not belong to any known phyla, but its DNA sequence was close to Cryptopsian lineage. It is widely distributed in coastal areas of Europe and North Atlantic. 2011, the algae was named Repermonads. It is a new candidate phylum. It is distributed widely in coastal areas of Europe and the Atlantic and the Pacific Ocean. So the last one is the strange phototrophic microorganism that were found in 1990 until 2011. It is a cyanobacterium-like particle UCYNA, which is detected using metagenomic analysis of nitrogen-fixing bacteria in marine water. This UCYNA contribute in the nitrogen cycle in pelagic water. But then this cyanobacteria lacks with photosystem 2, rubisco, the primary enzyme for fixing carbon dioxide and tricarboxylic acid. This UCYNA cyanobacteria might have a contribution in transformation of light energy into chemical energy. So after few research and after few discoveries, there is a reconsideration of photosynthesis. In the traditional times, the phototroph is thought to be the organisms that grow using light energy, while the photosynthetic organisms is the organisms that perform photosynthesis. This photosynthetic organism is the representative of the phototroph, and the primary producer is the organism that synthesizes organic carbon using inorganic carbon as their carbon source. In the other hand, in the modern times, the phototroph was defined as organisms that synthesize ATP by ATP synthase using transmembrane electrochemical potential of proton by pumping up protons from the membranous one side to energy. So that means that photosynthetic organisms are only part of phototroph. So please take note that primary producer is not only the organism that use light as their energy. The chemoautotroph organism is also a primary producer, even though it is in a small contribution. So that's all from me. Thank you.